All right, so it's already that time. We're ready for blood bank exam number 1. So we're going to review. Exam number 1 does cover lectures 1 through 6. Okay, so think of everything we've talked about. Talked about some immunology principles that we reviewed, some genetics involved in blood bank. We talked about determining your patient ABO. Talked about the DAT and the IAT. Okay, so that's what exam 1 will cover. As long as you guys can interpret your ABOs, as long as you guys understand the difference between a DAT and an IAT, and the characteristics of IgG versus IgM, you guys will do very well on this exam, okay? All right, so you're gonna have 86 questions. Each question's worth 1 point. You will have multiple choice matching, and there are some true and false questions on there. All right, so just remember some of our immunologic principles, the difference between intravascular hemolysis and extravascular hemolysis. Remember, intravascular hemolysis is usually associated when complement has been involved, and the complement cascade is activated, that C9 is on the cell membrane, punctures a hole in the red cell, and it causes immediate cell lysis, all right? Usually that's what we see when a patient receives the wrong type of ABO, all right? That would result in intravascular hemolysis. It is much more serious than extravascular hemolysis. Extravascular hemolysis usually occurs when the reticuloendothelial system is involved. So when the spleen takes out those cells, all right, that would be extravascular hemolysis. Again, not as serious as intravascular hemolysis. This comes to play a very important role when we start talking about transfusion reactions, okay? And if you think about the timing of symptoms of transfusion reactions, if it's intravascular, your patients are going to have immediate symptoms, all right? They're going to be complaining of flank pain, their fever is going to spike, tachycardia, hard time breathing, all right? Those are some symptoms of a transfusion reaction involving intravascular hemolysis. Versus extravascular hemolysis, the symptoms are much more lessened, all right? They might spike a fever, they might just, so they might have shortness of breath depending on the severity of it, but the extravascular is not as serious. Katie, did you have a question?
Yes. Would extravascular be delayed?
Yes, so oftentimes your patients, when we're thinking about transfusion reactions, usually if it's a delayed transfusion reaction, it's been 3 to 5 days since they received that unit of blood. And a reason for that is, and that's typically what we'll see in an amnestic response. So they actually already have the antibody in their circulation, but the titer is very low. And then once they're exposed to that antigen, that titer is going to jump up quick and start attacking those donor cells, and the spleen is going to take them out. I mean, that's why it's a delayed symptoms. Right, remember we talked about cellular immunity and humoral immunity. Make sure you guys are familiar. Again, this was just a review from immunology. So make sure you guys are familiar with what cells are involved in each of those types of immunity. For your cellular immunity, remember it is your macrophages and your T cells that are involved in that immunity. Your humoral immunity, remember, involves the B cells, where they will be transformed into plasma cells, and they will produce the antibodies. Okay, that's your humoral immunity. And remember, your T cells, those are your antigen presenting cells, okay? And you guys remember the structure of your immunoglobulin, all right? If you think about your antigen, all right, if it's on the red cell membrane or whatever foreign antigen might be exposed, whether it be on a bacteria, virus, the antigen has the epitope, all right? That's what causes your immune response. That's what will bind to the antibody. And when you think about your antibody, if you're looking at the IgG molecule, remember it's composed of 2 heavy chains and 2 light chains. For the IgG, the heavy chain is the gamma, and the light chain can either be lambda or kappa, all right? And so here's your FAB portion on the immunoglobulin molecule. For IgG, you have 2 FAB portions right here, right, at the terminal end of your heavy and light chain, okay? Remember, for your IgM molecule, your IgM molecule has 5 of these heavy and light chain combinations combined with a J chain. So the IgM molecule actually has 10 binding sites, right? That's what makes it so easy to overcome that zeta potential that we talk so much about. I want to make sure you guys understand in the primary immune response, let's say, for an example, a patient is given the wrong type of ABO, that is going to involve IgM antibodies, okay? They are usually the first ones to be involved in an immune response. Your IgG antibodies are more so involved in the amnestic or the secondary immune response, all right? So that's another difference between IgG and IgM. Not to say that IgM can't be this is most commonly what you'll see. Yes, ma'am.
So the IgM, those are more serious?
Yes. Yes, your well, when you're thinking about ABO, the IgMs are more serious. Now, we have some IgM antibodies that sometimes are considered, because they're room temp reacting, they are considered more of a nuisance in blood bank, because you really have to pay attention to how they're reacting before you say if they're clinically significant or more severe than the other. Right, so here's a example of the IgG and IgM right next to each other. Again, notice that your IgM is contains that J chain. Right, so here's your J chain in the middle, and then you have your immunoglobulin, your heavy and light chain combined by that J chain. And notice the 10 antigen binding sites on that IgM, okay? And your FC portion of your immunoglobulin, this is where complement will bind. And then remember that the heavy chains and light chains are joined together by disulfide bonds, all right? So right here in the middle area right here, this is a hinge region. That what gives the antibodies its flexibility. It can kind of fold, all right? All right, you guys make sure you understand the difference between sensitization and agglutination. We focus on both of these in blood bank. Given that the IgG molecule is so small, it might actually attach to the red cells. That indicates that the cell has been sensitized, all right? That's sensitization. However, we are unable to visualize that with our blood bank serological testing, okay? That is why we often have to use our anti-human globulin reagent that we've talked about, which will enable agglutination, right? Agglutination is when all the cells are brought together and we are able to visualize that antigen antibody complex bringing the cells together. That is agglutination, okay? Cross-linking is a key word there for agglutination, and that's what the key role of AHG is. All right, keep in mind your zeta potential. We talked a lot about that. Remember, the zeta potential is the negative charge around your red cells. It makes those cells naturally repel each other. So if we are trying to visualize when an antibody attaches to the red cell membrane, overcoming that zeta potential so we can visualize agglutination is very important. And that's why our ionic strength, our incubation temperatures, the saline we use, that's why blood bank incorporates all of those technologies to overcome that zeta potential. All right, make sure you guys remember prozone versus postzone. Remember, when we are thinking about the antigen antibody complex and we want the antibody to bind we want the antibody to bind to the antigen, we want to allow for that zone of equivalence, all right? We don't want prozone where we have too much antibody, and we don't want postzone where we have excess antigen, okay? So make sure you guys are familiar with that terminology, and make sure you guys understand both of these can cause a false negative, okay? If you don't have enough antibodies, then it won't bind to the antigen site on the red cells, therefore you'll have reduced amount of agglutination, right? That could result in a false negative. If you have too much antigen, all right, then you have too many antigen sites, and then your antibody is all bound up, and that could also result in a false negative, okay? Oh, sorry, Sarah. So Sarah asked, is sensitization the antigen-antibody complex? That is. So the antibody in sensitization, the antibody has already attached to the red cell membrane. Okay, think about your IgG molecule, how small it is. Before you add your AHG antisera, you're unable to see that binding because the IgG is so small, it's unable to form that lattice network of agglutination, all right? But even though the cells has been sensitized, we are unable to see that in our blood bank testing, okay? But the antigen-antibody complex has occurred. Typically in IgM, that's not a problem. IgM is so large, it can bind to multiple red cells, bring them together, and we will see that agglutination. All right, so typically sensitization is a concern with IgG because the cell has been sensitized, but we are unable to see it. All right, so we need AHG to form that bridge and bring those cells together. We need to help those antibodies overcome the zeta potential. We have some other methods that we can help overcome that zeta potential. Centrifugation, remember, so before we read any of our test tubes in blood bank, we will always centrifuge. There's always going to be a centrifuge step involved. Typically, you're going to be at 34 RPMs for 15 seconds if you're doing like your ABO reactions. If you start adding on some specific, more specialized antisera, that might even have a 30-second centrifugation speed or time. So we always want to check your package insert, but more commonly in blood bank, you're going to centrifuge for 15 seconds, okay? Does anybody know what over-centrifugation can cause if you spend longer than the suggested time? False positives, and the reason for that is if you spin too hard, your cell button is going to be very strong. And then it depends on your shaking technique as well. You could result in a very strong cell button, could give you some false positives or a stronger reaction than what really is there. On the opposite side of that, under centrifugation, or maybe you totally forgot to centrifuge, what would happen? False negatives, okay? If you're looking at your test tube results and you happen to notice a small pink tinge to your supernatant and a very small cell button, what does that indicate? Hemolysis. Remember, in blood bank, hemolysis is considered a positive reaction, okay? And so usually it's notated, if you are writing your results, you would want to notate an H. The letter H indicates the presence of hemolysis. That is indicated that the antigen-antibody complex has occurred and complement has been involved, right? Complement will result in that cell lysis. And then here again, we have our primary immune response involving IgM, IgG being involved in that secondary immune response as well. Notice too, in your primary versus secondary, your primary, like I mentioned, is going to be immediate, all right? Your secondary response is a more delayed reaction, okay? All right, so we talked about the ABO blood group system. We talked about our various types. Remember, Landsteiner first developed or discovered the various blood types in 1901. We have A, we have B, we have AB, and O. Remember, the blood type is determined by what antigen is on the red cell surface, okay? So if you are type A, that means you have the A antigen being expressed on your red cells. And if you're type B, you have the B antigen. And AB has both antigens. And then of course O is amorph, right? He doesn't have either the A or the B antigen being expressed, right? So that's why he's O. If a patient is given the wrong type of blood, I want you guys to understand that is a very severe occurrence. It could ultimately result in death, okay? So what's gonna happen is, remember Landsteiner's rule, for the antigen that you lack, you can build the antibody to. So if you have an O patient that's given A red cells, that O patient's anti-A is going to attack those donor A cells, all right? That will cause intravascular hemolysis. Cell fragments can get lodged in the kidney. Your patient's going to go into kidney failure, go into DIC. They're going to have a hard time breathing, all right? And that could ultimately result in death, all right? A little immunology review. Make sure you guys can differentiate between the phenotype and the genotype. So the phenotype is the observable expression, all right? So think about, if you're thinking about blood types, the phenotype would be type A. That's your phenotype. Your genotype could be AA or AO, all right? Given our blood bank serological testing techniques, we cannot determine the genotype, right? We can just determine the phenotype. You would have to do some molecular-based testing to see what gene is being expressed, all right, to determine if your patient is AA or OO. Now, if your patient's O, we know what their genotype is, right? They have to be OO because it is autosomal recessive in order to be an O patient, okay? If you guys remember, we also talked about the addition of the H or the H substance acting on the red cell. So remember, that's controlled by a gene, all right? So capital H, that's what it's designated as. If you are homozygous, big H, then you have the production of the H substance. If you're heterozygous, big H, you have the production of the H substance. And if you are homozygous, little h, you guys remember that they produce no H substance. So what the H substance does is add the L-fucose sugar onto your red cell membrane, onto that oligosaccharide chain that determines your ABO type, okay? So if you're H, homozygous, meaning you're producing no H substance, do you guys remember what the term for that type is? That's your Bombay type. All right, so it doesn't matter what gene is being expressed, whether they have the A and the B antigen, if there's no H substance, the N-acetylgalactamine sugar or the D-galactose sugar cannot be added on without that L-fucose. That's what makes the Bombay type so rare, okay? So the H substance has to add on that L-fucose in order for your A or B sugar to be added onto that oligosaccharide chain, okay? So if you are type A, that means you have N-acetylgalactosamine as your sugar, okay? If you are type B, that means you have D-galactose as your sugar being added onto that oligosaccharide chain, right? You guys remember that? That's a really good board of certification question. What sugars are associated with each blood type, right? And if you're AB, you will have both sugars being expressed along the red cell oligosaccharide membrane, okay? All right, so let's think about our Bombay type just for a minute. We established that our Bombay type is little h, no production of the H substance. All goes back to Landsteiner's rule, okay? If you lack the antigen, then you can build the antibody to it. So these patients that lack the H substance, guess what? They can build anti-H, right? That's why with these Bombay types, if you look at your chart that's in your lecture manual, the Bombay type reacts with all of your screening cells, right? That's because our screening cells are type O and have the H substance on their membrane. So your Bombay patient anti-H is reacting with those screening cells, okay? That's one way you can pick up on a Bombay type. They're gonna look like a type O patient, all right? And then their IAT is gonna be positive. And that's the reason why, is because they have that anti-H. With that being said, there's, depending on what blood type you are, will determine the amount of H being produced, okay? So if you are group O, and remember, if as the L-fucose sugar is added on, and then if you're type O, the N-acetylgalactosamine is added on, if you're B, the D-galactase is added on, that masks your L-fucose, okay? So if you're type A, then you will have the lower amount of H concentration, okay? If you're type O, because no additional sugar is being added on, you have the highest amount of H concentration. Okay, you guys need to know this, right? So O will have the greatest amount of H concentration, followed by A2, because remember, our subgroups of A are very decreased in antigenic, okay? So that's why A2 has the next highest amount of H substance. And then you'll have your B, and then A2B. A2B has a lower amount of H substance because you have both the A and the B sugar being added to the oligosaccharides on the cell membrane, okay? And then A1 and then A1B. Does that make sense to everybody? And again, here's your sugars, all right? So A is N-acetylgalactosamine, B is your D-galactose, and then the H gene controls the addition of that L-fucose. Remember, you have to have L-fucose in order for the N-acetylgalactosamine or your D-galactose to be added, okay? And remember, our ABO antibodies are of IgM classification. They are naturally occurring. So remember, your ABO antibodies they start developing about 6 months of age from the food you eat, the air you breathe, okay? They're naturally occurring. Versus our IgG antibodies, what are 2 ways that you can build IgG antibodies? What's the concern for females? Previous pregnancies, all right? And then there's one other, previous transfusions, okay? And the theory behind both of those is that you are being exposed to antigens that you are negative for. All right, so your body is going to recognize those antigens as foreign, and you're going to build an antibody to it. All right. Katie, did you have another question? No? Okay. All right, let's talk a little bit about A comma B. Anti-A comma B is a reagent that we use in our blood bank testing. It is not a combination of anti-A and anti-B. It is its own molecular structure, its own substance being produced. In group O patients, they will naturally produce anti-A comma B. Okay, so this is produced because they lack both the A and the B antigen, they will produce this. It can be found in their plasma. It is of IgG classification, all right? You guys need to know that for future reference. All right, we're gonna come back to that. Why do we use it as a reagent? Think about if you had a weak subgroup of A. Remember that we said weak subgroups of A often have a very weak antigenic expression. Sometimes with our blood bank testing, our reagents will not detect that. So you could have a group A patient with a negative reaction in your forward typing with your anti-A. And if your patient, what do we know if you are not A1, you're a subgroup of A? You could build an A1 antibody. Right? If you have an A1 antibody in your plasma, it will react with A1 cells in the reverse type, will look just like what type? Negative reactions in the front type, both positive reactions in the back type. That's a group O, all right? So we use anti-A comma B to try to pick up on any weak antigen expressions of those A. Sometimes you can even have, we haven't talked about any, but you can have subgroups of B. Very rare do you have subgroups of B, all right? We didn't discuss any in this class, but it can happen.
What would that look like? Would that look similar to subgroups of A?
The only thing, like a subgroup of B, it could have a very weak antigenic expression on the cells. I have never heard of a case where subgroups of B build anti-A. That hasn't been well documented. So I don't know, even though it's a subgroup of B, or if they could build antibodies, I guess, to one of the B molecule structures. That's what I meant to say. Not because they'll have anti-A, of course, but it's not well documented. Yeah. All right, but does everybody understand A comma B produced by group O? We use it to confirm group O's or make sure that our patients are not weak subgroups of A. That would be the main concern. And why is that a huge concern for donors? What happens if you type a weak subgroup of A as a group O? What recipient does that go to? O can only receive O, right? So if you give them a weak subgroup of A, then guess what? Their antibody is going to attack it. You just called a transfusion reaction. Okay, so that's why identifying weak subgroups of A is very important in donors. Okay, because we don't want it to be mistaken for an O, and then it's really an A, and it's going to go to an O recipient, right? That would be an incompatible ABO transfusion. All right, thinking about subgroups of A, about 80% of the population that is A is going to be A1, all right? Remember, when you think about the ability to build, think about an A2 patient can build anti-A1, think about your linear structure of the A1 molecule. Your A1 molecule contains linear and branched, okay? So here's your red cell membrane. You have the A1 molecule, branched, right? Very complex. You also have a very linear structure to that A1 molecule as well, okay? That's your A1, contains both of those. Your A2 only contains the linear portion. It is lacking this branched complex. And remember Landsteiner's rule? If you learn nothing else in this class, you will remember Landsteiner's rule. What you lack, you can build the antibody to. All right, so your A2 patients are lacking this molecular structure here. That is why A2 patients or other subgroups of A can build anti-A1, okay? Please do not ever tell me an A1 patient can build anti-A2. It is not possible because your A1 patients have the whole A molecular structure, okay? Does that make sense to everybody? Okay. I want you guys to understand too, I want to make this clear. Even though your patient is A2, that does not mean they will build anti-A1. Only about 1 to 8% of A2 patients will build anti-A1, okay? They can be happy A2 person living, no problems. The problem occurs when they're exposed to A1 red cells, all right? So if you're an A2 patient, you receive a transfusion from another A patient, but that patient happens to be A1, then you will develop that anti-A1 antibody, okay?
Would that be a delayed reaction since they don't have the antibody?
That would probably be a delayed reaction, and you're probably not going to have any side effects from that until next time BloodBank does your testing. You are going to have a positive reaction with the A1 cells in the reverse type. So it's not going to cause a transfusion reaction or anything like that. You're just going to build that antibody, and it's going to be in your circulation. So now, if you were to get exposed again to an A1 unit after you built that antibody, that's when the transfusion reaction will occur.
The immediate one.
Yeah, yeah. But for first exposure, you just built the antibody. So. And keep in mind too, that can happen in A1B patients as well, or your AB patients. AB patients can be A2B, all right? So if they're A2B, then they can also build anti-A1, all right? They can build it, they don't have to, okay? And remember, in the blood bank, we always want to try to use an EDTA specimen. However, we can use a clot tube. If that clot has already formed, we can use serum. But EDTA is preferred because if we ever need to do a DAT, on that patient, we want to make sure that EDTA chelates the calcium, right? And calcium can activate the coagulation, not coagulation, that just rolls off the tongue so easy. Complement cascade, all right? So we want to make sure that complement is involved in vivo, not something we initiate, all right? So that's why we would want to do the EDTA sample. We want to chelate that calcium. All right, and let's think about our ABO testing. Think about when somebody says forward typing, you are using patient red cells. And that's where your antigen is, okay? Forward typing patient red cells mixed with commercial antisera. That commercial antisera is your antibody, all right? If somebody says reverse typing or back typing, that is patient plasma. Your antibody is unknown, okay? We're mixing it with commercial red cells where the antigen is known, all right? So forward typing, we're looking for the antigen on your patient red cells. Back type, we're looking for the antibody in patient plasma, right? Does that make sense to everybody? Forwarding, reverse, front and back, is that terminology is used a lot in blood bank. Okay. All right, so how can you know if your patients are A1 or A2? What can we do? We can do testing with A1 lectin. It's derived from a plant, and it's called Dilocus biflorus. All right, so that's the plant it's derived from. It is an antibody. It is part of your forward testing. Okay, so you take the known A1 lectin, has A1 antibodies, mix it with your patient red cells.
We need to know the plant.
Probably a good idea to know the plant, because there are other, as we go on through the semester, there's other extracts for like M and N that we'll talk about. So make sure you know A1 lectin is Dilocus biflorus.
For this exam?
Yes. All right, so if your patient reacts with A1 lectin, what does that tell us? If you have a 3+, 4+ reaction with A1 lectin, they have the A1 antigen, they are type A1, right? If you have a negative reaction with A1 lectin, what does that tell us? Your patient is not A1. For this course purpose, we will call him A2, all right? Now, what other tests do we have to run when we use A1 lectin? A2 cells. What reaction do you expect for an A1 patient with A2 cells? Negative, all right? The purpose of A2 cells is to identify anti-A2, all right? So remember, our A patients have the whole A molecular structure, okay? So they're not going to build anti-A2. So they're not going to react with A2 cells, right? Remember, your A2 patients only have the linear structure of the A molecule, so we call them A2. They have the A2 antigen. They're not going to build anti-A2 either, okay? So they will not react with A2 cells in the reverse typing. Only types B and O will ever react with A2 cells, okay? Why will B react with A2 cells? Yeah, they lack the whole A1 antigen, so they're gonna build the antibody to it, right? Same thing with your O patients. They lack the antigen, so they're gonna build the antibody, right? That's why B and O only react with A2 cells. That is one of the hardest things for students in blood bank to wrap their head around. They want an A2 patient to react with A2 cells in the reverse type, all right? But if they're A2, they have the antigen on their red cell. They cannot build the antibody. Does that make sense to everybody? Okay. You guys spend some time as you're studying going over forward and reverse reactions. Make sure you can interpret straightforward types as well as discrepancies. And at the end of this review, I have added some examples that we're going to go through. All right. So please make sure you can do that. You will see several on this exam, or you will have ABO interpretations, I promise. And this table is actually in your lecture notes, and it goes through all the possible types. All right. And I just want to point out, notice that your typical ABO reactions should be 3+, 4+. They're very strong, right? And as you get into some of your ABO discrepancies, such as a A2B patient with anti-A1, notice that your antigenic expression of that A is a little bit weaker, right? You have that 3+ reaction there. And then even that discrepancy, Remember how you can identify your discrepant results. Those results are very weak, right? Oftentimes 1+ or 2+, okay? Also on here is an O patient with rouleau. So just doing his ABO-RH, since he's an O patient, you wouldn't pick up on that rouleau, all right? We don't see that rouleau until you get to your IAT, where that rouleau is being mistaken for agglutination. It's causing a positive reaction. And remember, the cause of rouleau, it's due to the excess protein in your patient's plasma. So anything you add patient plasma to will cause a false positive. Okay. If your patient was type A that had rouleau, your A cells in the reverse type would have a reaction in it. Okay, and that would alert you, hey, I have some type of discrepancy. Maybe I'm dealing with rouleau. You could look at it under the microscope and see the formation of a stack of coins. Okay.
How would you know that both patients have rouleau?
Just doing your ABO-RH, you're not going to know because he has that's expected agglutination in both A cells and B cells and A2 cells of the back type, right? You wouldn't pick it up until you performed your IAT. You're going to have a positive antibody screening. Yeah. That's one reason why for every patient that comes through BloodBank, we always do the type and the screen. All right, so every patient, if they potentially could be transfused, or the pregnant females, for the safety of the baby, we always do a type and screen, right? Every BloodBank patient will get a type and screen. Sometimes if autoimmune hemolytic anemia is suspected, then they might just order a DAT. And depending on your lab's policy, in order to do a DAT, they might also have to do an ABO-IAT for the full workup, especially if your DAT is positive. So, but everybody's going to get ABO-IAT, and we're going to add in an RH too. We'll do the RH on everybody. That's coming in the next module. Okay, so just a little bit of genetics related to BloodBank. Remember that your alleles are the alternate forms of the gene. So if you think about your chromosomes, there's specific loci on both of those chromosomes, okay? And that's your alleles. Remember, we talked about crossover. Crossover is the exchange of genetic material during meiosis. This comes into play because a lot of these red cell antigens are located very close together on the genes, and they are inherited as haplotypes. And so your M and N is a haplotype, your big S, little s is a haplotype, Duffy A and Duffy B haplotype. And as we start looking at our antigrams, that is how they are grouped together. And so they kind of run together in pairs. JKA and JKB inherited as a haplotype. And another word for haplotype is they're inherited together. Okay, so when I say inherited together, you could be positive or negative, right? You could be homozygous or heterozygous, depending on the gene expression. Remember, we talked about the genetics, so what determines the antigen expression on the cells. And so it is actually the DNA contained in the nucleus that will uncoil, and then messenger RNA is going to copy that DNA, and then it's going to travel to the ribosome, and it will line up with your triplets, all right? So your triplets are going to bring in amino acids, and that amino acid sequence will determine what protein is produced, all right? So that will determine what gene is being expressed on the cell membrane. We also talked about the Punnett square, demonstrating Mendel's law. Remember, you can use this to calculate frequencies. And so we will see this used in ABO. So if we have the genotype for mom and the genotype for dad, we can determine what the offspring could potentially be. All right, so ABO discrepancies. It's a good rule of thumb. What I always like to do, I break down when I'm looking at ABO interpretations, I break it down. I look at my forward type, all right? What am I front typing like? And then what am I back typing as, okay? If it agrees, you're good to go. So if you're front typing as an A, back typing as an A, your patient's A, you're good to go. No further investigation needed. If you're front typing as an A, back typing as an O, there's a discrepancy there. Something doesn't match. We have to figure out why we have an additional unexpected reaction, okay? And we have some examples that we're going to go through. It's very important, you guys, that if you encounter a discrepancy, it has to be resolved before you transfuse your patient. Now, in a trauma situation, if you're working up a patient, you encounter a discrepancy, OR is screaming at you for blood, you can give them O, all right? If that's the O is the universal donor, you can say, you know what, I'm still working on this patient's type, there's a problem, it's gonna be, 30 minutes. And they'll say, well, we can't wait that long. You can say, well, I can give you uncross-matched O units right now, all right? So sometimes they'll say, okay, yes, I want it. Sometimes say, well, I'll wait, all right? So it just depends. When you're working blood bank, you'll deal with physicians calling you freaking out all the time. And if you tell them what you can do, oftentimes they're like, well, we can wait, because they don't want to risk exposing the patient, to uncross-matched blood. So now if it's the ER or somewhere where your patient's bleeding out and they don't have a choice, they're going to do it.
So on the exam, are you going to tell us when we're trying to find like a discrepancy, if you're going to tell us whether it's a newborn or not? Like,
I don't necessarily know that I will tell you, but it'll be like listed in your options. So let's say in lab in the 9B, you guys had one of your problem patients where he had completely negative reactions in the front type, negative reactions in the back type. 2 scenarios for that. What could happen to cause that? It could be a newborn, all right, meaning that they have not built their ABO antibodies yet and we're not detecting them in the patient's plasma. That is the one reason we do not do reverse typing on newborns. Okay, it's just not routinely performed in blood bank. And that's because you just get negative reactions. If the baby has an antibody, it's maternal antibody. Okay, so usually if you're working at a hospital that does moms and babies, we always have to have mom's results as well. Okay, and we always correlate that. Usually when you receive a specimen, you'll go ahead and look up mom. And when a cord blood comes down from the labor and delivery, they usually have mom's sticker with the requisition as well. So you'll have mom's information too. So, all right, that would occur if you had a newborn. What's another reason? What if you had an older patient? Yeah, as patients age, the production of their antibodies decreases. So you might have a weak production of antibodies being produced there. What would you do in that scenario? What can we do to activate those IgM antibodies? We could put them in the refrigerator at 4 degrees, right? We could incubate them. Remember, IgM are room temp or cooler reacting temperatures. They love 4 degrees, right? So we could put them in the refrigerator for 15 to 20 minutes and then centrifuge and read for agglutination, okay? There's actually any type of immunocompromised patient. Maybe your patient's on chemotherapy. Their production of ABO antibodies might be decreased as well. So older patients, immunocompromised patients, when you'll see those weak antibodies in the back type.
Sarah just did a chat.
Yes, immunocompromised. And if you think about it, you could bulk newborns in that immunocompromised category as well, because they are considered immunocompromised until they start building their own antibodies and getting vaccinations. All right, you guys will need to be able to identify an autoantibody from an allo antibody, and you can do that by looking either at your auto control that is included in your IAT. All right, so we have some examples that we're going to go over on that, but one of the questions will be presented to you if you're given an IAT, is this a autoantibody or an allo antibody, okay? And remember, an autoantibody is one that you build to yourself All right, an alloantibody is an antibody that you have to other antigens. Okay. All right, so here's an example of an ABO discrepancy. If you have a 4+ reaction in the, with anti-A and a negative reaction with anti-B in the forward typing, 2+ reaction with your A1 cells and a 4+ reaction with your B cells, all right? Does your forward type match your back type? No. Which one of the results do you think is the discrepant? Which one do you think is the problem? A1 cells, because it has a 2+ reaction. Your weaker reaction is usually going to be the one that's causing the issue. All right, so what are some scenarios here? What could it be? It could be A2 with anti-A1. Could it be Rouleau?
Wouldn't you have to check that microscopically?
You would have to check it microscopically, but remember, this reaction is a true reaction with your B cells. All right, this reaction is the discrepant, but it could happen in any tube that you add plasma to. All right, so this could be Rouleau. What's the first thing you're going to do though? What would your next step be for this? You're going to run A1 lectin and A2 cells. All right, so what if I told you your A1 lectin was negative? What does that tell us? Is our patient A1 or A2? Patient is A2, and we expect a negative reaction with the A2 cells. That indicates our patient is indeed not group O, all right? And notice here, whenever you run A1 lectin, you always have to include controls. That is not done on a daily basis. That is done only when performed. Okay, so our controls checked out there. So how are we going to type our patient? A2 with anti-A1. You guys do not forget to describe this reaction right here. Okay, what is causing that reaction with the A1 cells? Most students will get that your patient's A2, but they forget to describe what's causing this reaction right here. This is how we know the patient has anti-A1. Okay, so you have to say my patient is A2 with anti-A1. Okay. All right, here's one. Our forward typing is 4+ with anti-A, 4+ with anti-B, but in our reverse typing, we have 2+ reaction with the A1 cells and the B cells. What do you guys think about this one? Rouleau, yes. So what would you do? What test could we do in BloodBank? Saline replacement. So we could take these 2 tubes, spin them down, remove the patient plasma. The thing you have to understand is that there was true agglutination here. The antibody has already attached to those red cells. Okay, that antigen-antibody complex has already occurred. So by removing the plasma, we are removing all that excess protein. Okay, and then we're going to add saline. We're going equal amounts. So however much plasma you remove, you add saline. So usually about 2 drops. All right, when you're dealing with that type. If your patient was anything other than AB, the true agglutination would still remain here, all right? But because our patient is AB, what reactions are these going to be after saline replacement? These are both going to go negative, all right? But I want you guys to understand true agglutination will remain, okay? So if your patient was type A and they had Rouleau, so you had an unexpected reaction with your A1 cells after saline replacement, agglutination in your B cells would remain. This tube with A1 would go negative if it was Rouleau. Okay?
So would it both be the same level?
Typically it will be, yeah. It's very common in Rouleau to see the same level. Versus the previous example, that one, you have a very strong 4+ reaction, right? So that makes you suspect this reaction is real, all right? This one is your problem, and then in Rouleau, if you have very weak reactions, both of them were the problem. And then if you were to look at Rouleau under the microscope, remember that it looks like a stack of coins. And in BloodBank, we can actually see that stack of coins when we look at the tube under the microscope. And a really good example, multiple myeloma patients will have this due to all of the excess protein, the excess production of immunoglobulins. All right, so let's talk about our DAT. You guys remember the difference between the DAT and the IAT. The DAT is your direct antiglobulin test. All right, we are detecting if an antibody or complement has attached to the red cells in vivo. Okay, so it's already happening inside the patient. We are not inducing it. We're detecting what has happened inside the patient. That's the biggest difference between the DAT and the IAT. Okay, the DAT detects what happened in vivo. All right. And remember the process of your DAT. We're going to start with polyspecific AHG, polyspecific anti-human globulin. Remember, it's termed polyspecific because it contains antibodies to both IgG and complement. If you have a positive reaction with polyspecific AHG, then you will perform your monospecific testing. Monospecific for IgG, monospecific for C3b, C3d, okay? And again, the reason for this AHG reaction or antisera, think about that IgG molecule attached to the red cell membrane. That IgG molecule is so small, it's unable to agglutinate to multiple red cells, form that lattice formation that enables us to see agglutination. Okay, IgG is incapable of that. So what the addition of the anti-IgG does is it forms a bridge, right? Your IgG or AHG reagent is going to bind to that antibody that's attached to a red cell. It's also going to bind to another antibody that's attached to another red cell, and it brings it all together, forming that lattice network so we can visualize that agglutination, okay? And don't forget, if you have hemolysis in your DAT tube, that could indicate complement. And then we're going to centrifuge our DAT, and then we will read for agglutination. There's no incubation involved in the DAT test. However, remember that we must wash, okay? We want to make sure we wash our patient cells very well. Why do we want to wash? Yes, we want to get rid of the excess protein that might be surrounding those patient cells, okay? But it doesn't cause false positives. It actually will cause false negatives. And you kind of have to think about that backwards, because what's going to happen? Inadequate washing has all this excess protein remaining around the cells. It can neutralize your AHG reagent. Once your AHG reagent becomes neutralized, it cannot bind to the antibody that is attaching to the red cells. Okay, and if AHG can't bind, guess what? We are unable to visualize agglutination, so we will have false negatives. Okay? Improper washing, failure to add AHG will result in false negatives. Okay? So Sarah asked, wash after centrifugation. So the process of your DAT, you're going to take about 2 drops of patient cells, and you're going to immediately wash them, right? You wash them as soon as the washing procedure is done, whether you do it manually or use a cell washer, then you're going to add 2 drops of AHG. Okay, so the AHG is added after washing. Right, just some reasons that we use the DAT. Number 1, if you're doing your IAT and your autocontrol is positive, that means that something on the patient's red cell is reacting with something in patient plasma. Okay, so if your autocontrol is positive, that is automatic DAT. Right? You can think of it as a reflex test in BloodBank. Okay? Anytime there is a mom and baby situation, if you receive a cord blood on a baby, we're always going to do a DAT just to investigate the possibility of hemolytic disease of the newborn, especially if that baby is in any type of respiratory distress, okay, or exhibiting symptoms of jaundice as well. Transfusion reaction, we will always do a DAT on your post specimen. So after the transfusion, a specimen will be collected. You're also going to pull that pre-transfusion specimen and do a DAT as well. Was that DAT positive before the transfusion? And maybe we just didn't pick up on something. Okay, that's part of our clerical check. Autoimmune hemolytic anemia, whether the patient has a warm autoantibody or a cold autoantibody, we will use the DAT to detect if something is coating patient cells. And then drug-induced reactions. All right, there are some drugs that can cause a positive DAT. All right, so now the IAT. Your IAT we use to detect antibodies in vitro. Okay, we're using patient plasma to see if they have any circulating unexpected antibodies. We're going to mix it with commercial screening cells or panel cells. All right, where the antigenic profile of those cells is known. Remember, both the DAT and the IAT use the same reagent. We're still using AHG. Okay, that anti-IgG will bind to the antibody that is either coating patient cells or in patient plasma. All right. Your IAT is what we call a 3-phase test, right? Remember, we're going to do initial spin. That's where we'll detect any of our IgM antibodies that are reacting at room temp. Then we're going to incubate at 37 degrees. And then we will wash and add AHG, and we'll do our AHG phase of testing. Okay, that's your 3 phase of testing with IAT. Now, as we move forward into specific antibodies react at different phases. And so that's one way that we can further do antibody identification. We can see what phases that they're reacting at. So that becomes very important as well. And again, we're detecting antibodies to the red cell antigens. Okay. And remember, the FDA mandates that on your screening cells and your panel cells, all of the red cell antigens that are known to cause clinically significant transfusion reactions, hemolytic disease of the newborn, they must be included on that antigenic profile. All right. And like we mentioned, your IgM antibodies will react at room temp and cold, and your IgG antibodies will react at 37 and AHG. Your screening cells are always going to be group O. And the reason for that is that, remember, group O lack the A and the B antigen. So think about you're mixing commercial red cells where your antigen is with patient plasma. What's also in your patient plasma other than unexpected red cell antibodies? You have your IgM, your ABO antibodies, right? So if you had any other cells other than O, those ABO antibodies from the patient would react with your commercial screening cells. That would give you positive reactions. You wouldn't be able to identify any other antibodies to other red cell antigens. Does that make sense? All right, so that's why they're group O, to remove the interference of ABO antibodies. Okay. We've talked about some false positives and false negatives as we have gone through the lecture, so keep those in mind. All right, so here's an example of an antigram. All right, notice this is the 2-cell screen. You might also see a 3-cell screen. Most manufacturers will either do a 2 or a 3-cell. What you guys need to know about this antigenic profile. Remember I said the FDA includes the antigen expression for all of the clinically significant antibodies, okay? So these are all antigens on your red cell membrane that have been known to cause like transfusion reactions or hemolytic disease of the newborns. So these are your antigenic expression of your red cells, all right, your commercial cells. This is how your patient reacted to those cells. Okay, so in this situation, notice that on screening cell 1, our patient was negative at initial spin, negative at 37 degrees, but a 2+ reaction at AHG, all right? And on screening cell 2, negative reaction at initial spin, negative reaction at 37, negative at AHG. And then you also have, this is your auto control, this third one right here. So patient cells mixed with patient plasma, and we have a negative reaction at all 3 phases of testing. So would you say this antibody is an allo or an auto? Definitely an allo. Do you think it's IgM or IgG? IgG. Do you think it is clinically significant? Yes, it is. Typically, if your antibody is reacting at AHG phase of testing, it is considered clinically significant. Okay. All right, so what I want you guys to understand is, let's look at screening cell. Sometimes this confuses students. This whole row, the top row labeled number 1, this is your screening cell 1. Okay. So if you're trying to figure out what antigen is present on the cells that could be causing this reaction in your patient plasma. Okay, the plus signs indicate the antigen is present. The 0 or the negative indicates that the antigen is absent. All right, so the screening cell number 1 is D positive, big C negative, little c positive. So screening cell 1 is homozygous for little c. Okay, big E negative, and this is the E, kind of hard to tell, little e positive. All right, so this cell is homozygous for little e. Okay. And then you can see this cell is homozygous N positive, negative for M, homozygous big S positive, negative for little s. So do you see how your haplotypes are positioned on your antigram? All right, Lewis A, Lewis B. With Lewis A or Lewis B, you're either positive for one or the other. You can't be positive for both. Okay. So screening cell number 1 is positive for Lewis A. Screening cell number 2 is positive for Lewis B. Notice that screening cell number 1 is heterozygous, positive for Kel and positive for Chelano. And then positive homozygous for Duffy B, negative for Duffy A. Homozygous positive for JKB and JKA. Does everybody understand homozygous versus heterozygous? So in the example, let's use Kel right here. This patient is heterozygous because if you think about the Kel family being inherited on the gene, remember you have 2 alleles, all right? So one allele is going to be positive for Kel, the other allele is going to be positive for Chelano. So he has both antigens being expressed. Versus Duffy A, all right, both alleles are expressing Duffy B, right? Neither of the alleles are expressing Duffy A. All right, so this patient is Duffy A negative. He lacks the Duffy A antigen on his red cells. So the way you can approach these IATs is, what antigen is present on screening cell number 1, but not on screening cell 2, that would cause this positive reaction in our patient plasma? That's what you have to ask yourself. All right, a lot of students get confused. This is the antigenic expression on your cells. This is how my patient reacted with those cells. This is your patient plasma. All right, so you can go through here and pick out what's present on screening cell 1, but not on screening cell 2. Okay, so would big E be a candidate? No, because big E is the opposite, right? Negative on screening cell 1, positive on screening cell 2. That doesn't match our pattern of reactivity, correct? We want it to be positive on screening cell 1. What about little c? Little c is definitely a candidate, all right? What about N? P1? Lewis A? Cal? What do you think about Duffy B? Typically, if it was Duffy B, you would have a reaction because he's present on both screening cell 1 and 2. You would have a reaction in your patient plasma on both cells as well. All right. That doesn't completely rule him out, though. All right. We have to keep dosage in mind, that as well. All right. But more than likely, it's not going to be Duffy B. What about JKA? Well, what about JKB? He's definitely a possibility. Okay. So there's your possibilities. Does anybody want to review that again? Does that make sense to everybody? We have, what's your next step? Not the elution. So if you had a positive DAT, then you would do the elution. If you had a positive IAT, is this confirmatory? Have we definitively identified the patient's antibody? No. What must we do next to confirm which one of these is the patient's antibody? Not molecular. We must run a panel. All right, so this is just either a 2-cell screen or a 3-cell screen. Keyword is screen, okay? When we do a panel, you guys haven't seen those yet, it's usually a series of more cells, all right? Usually, typically it's gonna be an 11-cell panel where you have more cells with more antigenic expressions on them for further definitive identification of the antibody. I mean, we'll get into why that is. You have to have a 95% confidence level. And so we'll talk about that when we start into antibody identification. So a panel would be your next step after a positive IAT. Okay. All right, here's another one. Notice that this one's actually a 3-cell screen, right? So it has screening cell 1, 2, and 3. And notice, here's your antigenic expression on each cell. Notice that screening cell 1 is negative. Screening cell 2 has a 2+ reaction at AHG, and then screening cell 3 has a 2+ at AHG. And also, what do you guys think about your auto control? So is this an allo antibody or an auto antibody? Auto antibody. That doesn't make sense. Okay. If you have an auto antibody, he reacts with everything. Okay, so if you have an auto antibody, screening cell 1 is going to be positive as well. All right. So let's pretend our auto control is negative. All right. I'm going to fix it. There we go. All right. Our auto control is negative. All right, it's probably hard for you guys to do. So what antigen is present on screening cell 2 and 3, but not on 1? What are our possibilities here? Should have printed these out for you. All right, little c. Take a look at Chelano. Somebody said Chelano. Notice that Chelano is actually positive on screening cell 1 as well. Probably not Chelano. Duffy B. Sorry, Aaron, you said that. What's next? JKB. N. Little s. Any others? No, so those are our possible antibodies. Are there a combination of any two that might work? Whenever you have multiple cells that are positive, right, if you have multiple reactions here, you always want to think about combinations as well. So it might not be one single antibody, it might be multiple antibodies. I don't think there's any combinations.
What would that look like?
So remember when we were doing our IAT lecture, we had an IAT example where screening cell 1 and 2 were positive, and screening cell 1 was positive for big C, and screening cell 2 was positive for big E. So it could have been a combination of big C and big E. They like to run together. Sometimes you'll see them together. But in this case, screening cell 3 is big E negative. So in this case, there's not a combination of any antibodies that would work. All right, would you consider this antibody clinically significant? Yes. Is this antibody IgM or IgG? IgG. Allo or auto? So allo. Can you determine your patient's blood type based on this information? No. Should check cells have been added to screening cell number 1? You guys remember what the purpose of check cells is? Remember, check cells are group O D positive cells that have been coded with anti-D. Anti-D is an IgG antibody. So think of check cells as IgG coded cells. So what's the purpose of AHG? It binds to IgG antibodies, right? So if you are doing an AHG test and you get a negative reaction and you have added AHG, you appropriately washed, then you have unbound AHG in that tube, right? When you add check cells are coated with IgG, all right? When you add check cells to the unbound AHG, spin and read for agglutination, you are going to have agglutination, all right? Remember we talked about check cells being an internal validity test, all right? So it confirms negative AHGs. You must always, always use check cells whenever you're using AHG, all right? So should you add check cells to screening cell 1? Yes. Should you add check cells to, dang, that's yes. I must have been asleep when I did this. Add those backwards. Okay. Should check cells be added to screening cell 1? Yes. What about 2 and 3? No, because they're all already positive. Okay. If you have agglutination after adding AHG, whether macroscopically or microscopically, you do not have to add check cells. Okay. You're done. You call it positive. Keep in mind, when you add check cells, that is not a representation of your patient's reaction. Right? It's just an internal quality control that blood bankers do to make sure, number 1, washing was appropriate, and number 2, that AHG was indeed added. Okay? All right, so let's go through some sample questions. Which blood group has the most H substance? O. Anti-A and anti-B are produced due to what? What classification of antibodies are those? And then how do we build them? We don't have the antigens. And remember, these are ABO antibodies. They are naturally occurring. So it's from the air we breathe, the food we eat, okay? Why do we wash the cells for AHG testing? To get rid of the unbound protein. If you have unbound protein, that excess protein can neutralize your AHG reagent, could cause false negatives, okay? And make sure you guys review those ABO interpretations. But what is the zeta potential? It's a negative charge around your red cells that cause them to naturally repel each other. It's a huge concern for us in blood bank. We have to overcome that zeta potential. Will there always be agglutination if an antibody attaches to a red cell antigen? No. It could have, the antigen antibody complex could have occurred, such as in an IgG antibody, that is sensitization, right? We are unable to see that in blood bank. All right, so what's your prozone? Excess antibody or excess antigen? That's antibody. What part of the antibody attaches to the antigen? That's your FAB region, okay? If more cells are added to your test tube than drops of antisera, so you're adding more cells, is this prozone or postzone? That's postzone. Because you have two, you're adding more cells, that's where your antigen is. So that would be excess antigen. If you have excess antigen, what does that cause? False positive or false negative? False negative. Why do we centrifuge? Overcome zeta potential. Centrifugation will push those cells together, allowing for the antigen antibody to combine. If you shake the cell button too hard, will that cause a false negative or a false positive? False negative. Shaking too hard, the more you shake, the more that cell button breaks up, all right? And if you shake it too hard, you could have a false negative. Okay, that's why shaking technique is so important. What if you had too heavy of a cell suspension? What's in your cell suspension? That's where your cells are. That's where your antigen is. So if you have too much antigen, what is that? That's a false negative. So you guys really have to ask yourself, where is my antigen and where is my antibody? Okay, both too much antibody and too much antigen will cause false negatives. Both of them will cause false negatives. Okay, what are check cells? They're cells, they're group O, Rh positive, so they have the D antigen coded with anti-D. All right, so that's an IgG antibody, so they're IgG coded cells. Remember, we use them to confirm all negative, both macroscopically and microscopically, AHG tubes, okay? What's your most common blood types? A and O, right? The majority of the population is going to be O and A. How are most blood group antigens inherited? Genetically, but is it autosomal dominant, autosomal recessive, autosomal codominant? Codominant. When do we add check cells? Every time you have a negative AHG result, you will confirm that negative with check cells, right? It's going to make your tube go positive. That ensures that AHG has been added, okay? All right, so here's an example of Punnett square where you can determine the genotype of your offspring. So what's your phenotype of your AO patients? If this is your genotype, what's their phenotype? They're O. What's your phenotype for homozygous, or sorry, heterozygous? O. O. Okay. Remember, in order to be O, you have to have both the O alleles lacking A and B expression. Okay. Remember, type O is an amorph, right? It's not really an O antigen. It's just that the patient lacks both the A and the B. Okay. Can you determine the exact antibody just by your screening cells? No. Have to make, we can theorize, right? We can say these are your potential antibodies, but we must perform a panel, all right? What blood type of our screening cells? O. And what does a titer mean? So your titer is the concentration of your antibody, all right? A huge concern in blood bank, especially for amnestic responses. If that, especially let's say you're dealing with a JKA or a JKB antibody, and that titer drops down to undetectable levels, we miss that antibody in blood bank, we give that patient a JKA or B positive unit, we cause them to have a delayed transfusion reaction, okay? Don't confuse titer with elution. What's the process of elution? Well, in a way, if you think about it, but let's say you have a positive DAT, what's your next step? To perform an elution, right? So if you have a positive DAT, you know something is coding patient cells, whether it be if you have a positive reaction with your monospecific IgG, then it's an IgG antibody, right? We want to do an elution. What an elution is going to do, you can do a heat treatment, you can do an acid treatment. It will dissociate that bond from the antigen antibody. So it's going to pull off that antibody from the red cells. Now your antibody is in your supernatant. We can run our supernatant just like we did with the patient plasma using our screening cells. We can do that with the eluate, right? And we can identify what exactly is coding patient cells, right? That's the process of elution. We'll talk more about that later too. Right, which cell recognizes antigen and regulates the immune response? T cells or B cells? That's your T cells. Which cell produces your antibodies? That's your B cells. Or B cells transforming the plasma cells. What does sensitization mean? That's right. The antibody is attached to the red cells. However, it has been unable to completely overcome that zeta potential. We cannot see that sensitization, all right? In BloodBank, are we only interested in agglutination? No, we care a lot about sensitization. That's why we have our specialized reagent AHG, all right? What specimen do we use for ABO typing? We want an EDTA sample, and remember, we will use both patient red cells and patient plasma. We have to have both. All right, and remember we talked about protein synthesis, which controls which antigens are expressed on the red cell. Remember, that is controlled by the DNA in the nucleus, which will control the gene expression and determine what antigen is expressed on the red cell. It's all nucleotide-based pairing, right, with a template made from that nuclear DNA. Will A2 plasma react with A2 cells? No. Nobody better miss that question, okay? Is anti-A, B made from an AB patient? What patient's it made from? Made from group O. What happens if a group O person receives blood from a group A? Transfusion reaction. So what's going to happen is your anti-A in your O person is going to attack those A donor cells, all right? That will cause a hemolytic transfusion reaction, could ultimately be fatal. Is complement a protein? When do we use lectin? When there's a discrepancy, right? When you expect your patient is a subgroup of A and has built anti-A1, all right? Do most A2 people build anti-A1? No. Again, only if they're exposed. So you have all these A2 people out there that don't know they're A2, okay? They're happy. Those questions I asked when we were reviewing the IATs, make sure you can answer those. Is it an auto antibody? Is it an allo antibody? IgM, IgG? What are your possible antibodies? Okay, so match your pattern of reactivity. Make sure you understand that is not a definite ID of the antibody. You have to do a panel, all right? And it does not, unless you include it on the antigram in your patient information, you do not know your patient's blood type, all right? You know your screening cell type, they're all O, but you don't know your patient's type. And then ask yourself, where's my antibody, where's my antigen, all right? Make sure you remember that. Red cells have the antigen, plasma or commercial antisera has the antibody.
Do you do your ABO typing before you do your screen?
So usually in BloodBank, we're always concerned about turnaround times because usually we're dealing with patients that need transfusions or have been involved in an accident or in the OR and they need transfusions quick. So we always try to do our tests most efficiently. How can we get this result out the quickest? So you're going to start your IAT first because it has a 15-minute incubation at 37 degrees, right? So you're going to get that started while you're incubating, then you're going to do your patient's ABO-RH. All right, so you'll be done with your patient ABO-RH. You'll be able to report out your IAT. If there's no problems, that patient is good. You can cross-match units, ready for transfusion. All right, and then also by setting up that IAT first, you're quicker to know if there's any antibodies in your patient plasma. Sometimes these workups are very complex, especially if you're dealing with multiple antibodies, especially if your DAT is positive, then you have to do an elution. That gets very time consuming. It is not uncommon for patients in BloodBank to have a 3 or 4 hour workup. So depending on what all has to be done. And as we build on that, you guys will see. Especially if you're dealing with a warm auto. I told you guys those are nightmares for BloodBanks. I'm not kidding when I say that, especially if you have to do auto absorptions. That takes time, and sometimes you have to do 4 to 5 treatments before that auto, and then it's not even absorbed out, it's still reacting. So it depends. But the first thing you want to do when you're working up your patients is get that IAT going. Now you guys have, there's 2 ways, 3 actually, You can do the tube. So the way you guys have been doing ABOs so far, that's the tube method. There's another method that's on the market now. It's called the gel method. And you'll use these little cards. They have columns in them, and the reagents are already added in the columns. And so then all you have to do is add your patient sample, incubate, read that. It's called AHG, phase of testing, for your IAT. It removes the initial spin, removes your 37. It gets rid of all the nuisance antibodies that react that initial spin. So a lot of places are using gel. There's also, you guys will get to see a demonstration from Immucor. They have their own blood bank testing technique. It's called the Capture, and Capture R is for their ABO. It uses the ELISA testing. It uses the little wells. And so used to be Ariel is going to come make a demonstration. Now there's a new vendor, a new representative. She's going to come, it's at the end of the semester. You guys will actually get to do that. She'll bring in her instrument and let you guys all do that. So depending on what hospital you work at, there might be different technology used. However, the tube method is the gold standard. Okay, you can always reflect back to the tube method. All right. All right, so let's play, and then we're almost done. I know you guys are tired of sitting here. All right, so what's my blood type? All right, 4+ reaction with anti-A, negative anti-B, 4+ with A comma B, 4+ A1 lectin, negative A1 cells, 4+ B cells, and then screening cell 1, 2, and autocontrol or negative. What's your type? Yes, A1 with a negative IAT, right? What about this one? 4+ anti-A, negative anti-B, 4+ anti-B, negative with A1 lectin, 4+ with B cells, and then all the rest of your reactions are negative. That's A2 with a negative IAT. What about this guy? B. Why did he react with A2 cells? So is that what you expected? Yes. What about this guy? O with a negative IAT. What's this guy? A2B. What's this guy? I heard A2B, A1B with the negative IAT. What about this guy? Say it louder. A2B with the anti-A1 with a negative IAT. Okay. Does everybody see that? Everybody pick up on that anti-A1. How do you know he's A2? The A1 lectin is negative. What about this guy? A2 with anti-A1. What about this guy? What do you think his type is? A1B. So what would you do? Saline replacement. Now what's his type? A1B with a negative IAT after saline replacement.
Could you indicate probable reload?
You would resolve it. You would perform saline replacement, and depending on if you have a blood bank system where you enter your results in the computer, you'll have truth tables built in. You would want to enter these results. You want to enter what you have. Truth table is going to pop up and say, this is not a valid type, something's going on. You would have to then put a note in saying saline replacement performed, and then you could enter these results. And then you could say A2B patient with Brulo.
In that case, you wouldn't have to to necessarily say, I saw it in the microscope too, or
No, you could just say rouleau present. Yeah.
just based on these two?
Now you could, this could also be a cold reacting antibody. Maybe the patient has, but probably if you have a cold reacting antibody, I don't know if it'd be on both of these cells, you might have a negative in there. If any of these were negative, then it's not rouleau. All right, so rouleau, remember, is going to happen everywhere you add patient plasma to. All right, so you could potentially have a cold reacting autoantibody too, right, because your autocontrol is positive. So if you're thinking, oh, is this rouleau or an autoantibody, you could look under the scope, see the stack of coins and think, I need to saline replacement, and then you would be done. As long as your IAT went negative. If you do saline replacement and you still have agglutination, then that means there's an antibody there too. Okay. All right, anybody have any questions?
Is the reaction with A1 cells always weak, like 1+ or 2+?
If you're dealing with a discrepancy, yeah, it'll always be weaker. And that's just because it's the patient's immune system building those antibodies. It does not react as strong as our commercially manufactured antisera. All right, so you see these anti-A, anti-B, anti-A B, these are manufactured, right? Remember using the monoclonal method, all right? So they're very highly specific to a specific epitope, all right? So your patient's antibody is not produced near as strong as your commercial antisera. And that's why it's usually always, that's not a good example. Like that, 1+ or 2+, very common when we're dealing with discrepancies.
And then do you know if a patient has an auto antibody versus an allo only based on if you do the auto control?
Right. If your auto control is positive, we call it an auto antibody. Yeah. And then it's our job to figure out what type of auto.
Because I was on the solo, one of the questions was if it's an allo or auto, and I wasn't sure because like an auto control wasn't added. So like I wasn't sure.
Huh, let me check. I wonder if the auto control, was it left off? Let me see if there, what details were given.
Okay.
So in the first IAT, screening cell 1, but not on screening cell 2. Okay, if you look at the first example, Sarah, notice screening cell 1 is where your patient reacted. You have a reaction in patient plasma, right? So something on screening cell 1 is causing that reaction. Notice on screening cell 2, it is negative. Yeah. On the second example, it's kind of the opposite. Screening cell 1 is negative, screening cell 2 and 3 are positive. So the antigen has to be present on both cells 2 and 3 to cause this reaction. Does that help? Sarah brings up a very good point. I want you guys to hear this really quick. If at any phase you have a positive reaction, initial spin 37 AHG, your IAT is interpreted as positive. Okay? If you are dealing with an IgM antibody that reacts at initial spin, it's very common for IgM antibodies to react at initial spin and then go away at 37 in AHG. Right? So you would have reactions at initial spin, and then you would have negative reactions at 37 in AHG. That is still interpreted as a positive IAT. All right. So if you have a positive reaction in any phase of testing. All right. So if you had positive reactions at initial spin, and I asked you if this was an IgM antibody or an IgG antibody, what would you say? IgM. All right. If it's reacting at initial spin only, then it's an IgM antibody. Okay. But if it's positive at any of these phases, it's considered a positive IAT. Okay. I mean, you guys get plenty of practice as the semester goes on. From here on out, we will always do these. Okay. This is the first step. Again, this is the screen on every patient.
So if it's an autoantibody, it would be positive at initial spin 37 and 8.
Probably. But if it's a cold reacting warm autoantibody, you would see it reacting at initial spin. It's possible for cold reacting autoantibodies to go away after warming at 37 degrees. And then it's possible to see warm antibodies react at initial spin 37 and AHG. It's also possible to only have warm autoantibodies, where you'll be negative at initial spin, negative at 37, and then positive at AHG only. So those autoantibodies are tricky. But you always have to look at your auto control and what temperature is it reacting at. So if it's only reacting at initial spin and your auto control is positive, it's a cold reacting IgM autoantibody. Okay. If it's reacting at AHG, then it's a warm reacting autoantibody. You guys don't have to worry about that so much on this exam. That's a little bit further down the road. But I wanted you guys to be able to interpret your IAT. So if you have any positive reaction in here, irregardless of what phase it is, it's reported, it's interpreted as a positive IAT. You have to do a panel. And don't be confused by check cells. If you're given check cells results, I always tell students, cover those check cell results up. Put a piece of paper over them. You know, whatever you need to do, don't let them be mistaken for patient results. Okay? Because you're going to have all positive results on your check cells. After adding check cells, it's going to make your tube go positive. All right, so a good rule of thumb is to cover those check cells up. Don't let them confuse you.
Yeah, girl, you can have. You know, all really? Wow.
Yes, it's dependent upon the phase. I'm glad you asked that question because I wanted to tell everybody that.
Every time I hear like make sure you know, like you know how she gets crazy. She's like, shh. And I'm like, where we going?
BloodBank's like riding a bicycle. It'll all come back to you. Okay, if you guys have any questions as you're studying, please let me know. Happy to answer them. Oh, remember, you guys can take the exam on Monday using lockdown browser anytime. Okay, I'm not going to proctor it for you guys.
Oh, sure. Jenny told me it's allo because it only reacted with the B cells and not the A cells. That's how you know?
Yes.
Okay. So auto would react with auto.
Yeah, so if it was an autoantibody, you would have a reaction with your A cells.
Okay.
The key thing to note about autoantibodies is not only your auto control going to be positive, but you're going to have pan reactivity. You're going to have positive reactions across the board. Anything you add patient plasma to, it's kind of like Rouleau.
I don't understand, like, if you're not looking at that and you're just looking at, you know, the charts you make for the ADO, like, which one is it? How do you tell from those that it's an auto or allo?
Like this one.
I know lecture 4 had one. I was trying to figure out, so I was looking at the example you gave and in that chart, and I didn't even.
So this one doesn't give you any details about phases of reactivity. It just tells you that screening cell 1 was negative, screening cell 2 is negative, and your auto control is negative. So while this says like B with a cold auto antibody, they're giving you that it's anti-I. Just looking at this, you don't know what phases of reactivity it is. You have to have each phase of reactivity.
What do you mean by that? Like phase?
initial spin 37 AHG.
Okay, so you have to do.
Yeah, you have to. The point of this is just to let you know that you will have positive reactions. But you really need further details. If I'm ever asking you to tell me if it's an allo or an auto, IgM or IgG, I will give you all phases. Yeah. Just based on this, they're just telling you kind of what to expect. So there's some language in BloodBank, phase of reactivity, pattern of reactivity. So when you're interpreting IAT or panels, we always look at initial spin reactions, 37, and AHG. That's your phase of reactivity. Your pattern of reactivity this is true both for your antigram and your panel. Pattern of reactivity means that your patient reactions match the antigenic expression on each cell. So for like, we use our pattern of reactivity to identify what antibody it is, basically. So we know screening cell 2 and 3, the antigen is present. So it could be little c or WB. That's using our pattern of reactivity. And then as we get further in, we'll start looking at strengths of reaction as well. So you might have some 1+, 2+, and 3+ over here. They might not all be the same reaction strength. That could indicate dosage. That could also indicate multiple antibodies present. And so that kind of helps you identify if there's more than one thing going on in that patient.
Okay. And then whenever you're trying to figure out like a combo of two, what's the word, like C and E.
Mm-hmm.
So like for this one, what would C and E look like to make it?
So if you had big C and big E?
Yeah.
Okay, so take a look. Big C is on screening cell 1. Big E is on screening cell 2. Both are negative on screening cell 3. So for that combo to occur, you have a positive reaction in your patient plasma with screening cells 1 and 2.
Okay. So what if, like for this one to fit or whatever, would it for C, could C screening cell 2 be positive, and then for big E, I guess the very bottom one be positive?
For little e,
For big E.
big E, screening cell 3 would not be positive at all because big C and big E are both negative. Does that make sense? So if the antigen is not present on the cell, then there's nothing to cause a reaction in your patient plasma.
Okay.
Does that make sense? It will make much more sense when we start doing panels. We use our negative cells to rule out. And so you guys will be taught the process of ruling out. And that's where you go through each cell one by one. For every negative result you have in patient plasma, you can do what we call a cross-off procedure, and you can eliminate antigens. And that's another reason why the panel is more diagnostic than the IgG. I was trying to think of a good combination for you. You couldn't have both big C and, well, you couldn't have both anti-big C and little c. You can't be negative for both. So.
Is it, so can you just make combinations with any of the ones in the...
Oh, here's a good combination. All right, let's say you had a positive reaction on screening cells 1, 2, and 3, but your autocontrol is negative. Okay, with that autocontrol being negative, you know you're not dealing with an auto antibody, you're dealing with an allo antibody. Right. So it could be a combination of little e. All right, take a look at little e right here. He's positive on screening cells 1 and 3, and Kel is positive on screening cell 2.
Okay.
That would be a combination where all 3 cells are positive, but your autocontrol is negative. So you know you're dealing with something that's either present on all 3 cells or a combination of antibodies.
Thank you.
Anybody else have any questions? Hi, Sarah.
And the new